Hello, everybody. Welcome back to another episode of the Lil Dudes Insect Academy podcast. Wait, before we get started, I just wanted to say thank you for tuning into this episode of the Lil Dudes Insect Academy podcast and just ask you to please hit the like button below or click the subscribe button if you're watching on YouTube or follow this podcast if you are listening. That really helps the show out. But also, if you want to help us further, then join our Patreon. We have an awesome community that is growing there. Or if you want to make a one-time donation, we are a nonprofit foundation, so head to our website. Both the Patreon and the website will be linked down below. But without further ado, back to the episode. I'm your host, Bradon Coy, and I am super excited today because we have another guest on the show. Her name's Krisa Ye, and I'm super excited to have her on because she is a postdoc student studying ants at the University of Stanford. And I am super stoked to chat with her about her research, and let's just jump right into it. Go ahead and introduce yourself for us, and we'll jump right into it. Thanks for coming.
Hi, thank you, Brandon. Hello, everyone. I'm Krisa Ye. I'm originally from China, but now I'm in the U.S. I'm currently a postdoc in Stanford study ants, and I'm really happy to share you about my stories in majorly study ants and a little bit about fruit flies. Yeah.
Awesome. So, yeah, let's jump right into it. You did mention that you also worked on fruit flies, but ants are like your passion, like you're most interested about ants. What is so interesting to you about ants? I mean, I love ants too. They are kind of what I'm thinking about studying in the future, but what gets you excited about ants specifically?
So I think one obvious reasoning is more like I think every kids get that, or when adults are little kids, they have that period of times, like just looking at the ants doing things on the floor and trying to figure out what they're doing. Sometimes they are carrying food, going back to the nest, or sometimes they generate some really interesting but mysterious patterns on the floor, and then trying to disturb them. And sometimes the ants even attack you, that kind of stuff. Like one of my really early memories, like around 3 years old or something, was a big red ants biting on my toes, and I remember it was painful. I don't even remember why they are attacking me, but that's one of the part. But I wouldn't say that is the reason why I want to study ants. I think I got into ant study. It's kind of by accident. It was when I was undergrad in Illinois. I was originally from China. I did 2 years of my college in China. I was doing majorly actually in plants rather than animals because my college is more focused on plant study. But during that time, I got classes about animal behaviors and evolutions. But unfortunately, those are not really the focus of the university. So it was some professors, they study plants, but they're interested in those topics and they gave in the talk, but they're not really focused on those studies. So they are more like, oh, we are studying this with you guys and we are reading this textbook together.
Right.
But that really got me into the topic about animal behavior and the evolution of animal behaviors. And I know I couldn't study those when I was in China in that university. So that's the reasoning why I come to the US. So it was driven by more on the evolutionary biology and animal behaviors. But to be honest, I had no idea how exactly people doing those studies, what are the options. So for example, when I was thinking animal behaviors, when I was at undergrad, I was thinking more about maybe looking at some zebras and lions in Africa, or some like a bird's behaviors, because we saw a lot of like birds like a language or learning behaviors, or some neurobiology studies in mouse and rats.
Yeah.
But when I was in Illinois, starting my first year as a transfer student, and then I was like, I heard people saying it's time for undergrads can get a chance to go to different labs to figure out what they want to study, maybe start independent projects and stuff. So I asked my department people, like I was in integrative biology. When I was joining there, I also had no idea what is integrative biology do, but basically it's evolutionary and ecology biology rather than molecular and cellular biology. So it's exactly what things I want. So people in my department told me, oh, they have all kinds of labs. There are bird labs, there are rats labs, and there are also honeybee labs and fish labs, and there's ants lab. So for me, it's like, oh, I didn't even know people study like ants and honeybees, what they are study for. I was also equally interested in bird study or fish study because I just never got experience from those. So I said, okay, I will just send several emails to those different labs and see how it goes. And I think that was just kind of randomly, I sent my first email to the ants lab. And I said, okay, I will wait for a week or two, and then if the PI didn't respond, I will send another email to remind him, and I will send another email maybe to the honeybee lab. But that professor, Andy Flores, I guess he's like a really fast action person, so he sent like a response to me like days after. She said, yeah, we're recruiting undergrads. If you're interested, come to have a look. We have all kind of cool studies going here. So I went to his lab. During that time, most of the people in the lab are actually not in the lab. They are all in the field because that lab is also focused on field research. There was a graduate student, Josh, who I think now is already graduated and still in the ant field. So he showed me the ant room that they have some of the most amazing ants. Like this, they are majorly focused on trapjaw ants, which is the ants with really long mandibles. And they do this amazing high-speed mandible snappings. So sometimes, like when I look at the ants in the ants room, I don't even really know what's happening because you will just suddenly hear a snap, and then there's like a little tiny thing just flipping in the air. So that was actually the ants doing the snapping, and it's so fast, and then they're causing this motions for the ants to jump in behind and stuff. And that was Josh's major projects. Like during the time he was looking at the dynamics of the snapping of the ants, like he's comparing like the snapping dynamics, like the speed of velocities or the strengths of the ants go with snapping, compare like relevant to their different body size, because those ants have different body size, and also like how strong the snapping can cause in the ants moving around and stuff, which is really fun. So my first project about ants was helping Josh to tracking these high-speed motions frame by frame, because we use high-speed cameras to capture the snapping behavior, and then I was tracking the points on the snaps, the mandibles, to see the trajectory of the mandibles, and then by using that we can calculate the velocities or the strengths the snapping is causing. I was doing that for like 2 years, and I found it was really fun. Besides, like we carry like a big box of the snapping ants going to like the microscope and like a recording rooms with those fancy speed cameras. We also need to feed the ants because we are the only 2 people in the labs during that time. I think there are other undergrads, but usually like at different, like usually I'm the only one working with Josh. I guess other people at different timeline, but every week I need to feed the ants. And feeding the ants, it was really also fun. Is one thing is I work with the ants, so I kind of know, oh, these are my colonies, and we work together. I feel like I'm kind of know them. So that was the time I actually can have some not working time, but you can say like a quality time with them. Like I give them food, but help them to clean up the colonies, and
those colonies are very small, right? In terms of ants.
Yeah, there are not many of them. It's just like in a small, like a food container, that kind of things. And you can also see the ants coming, because you usually put a nest inside of a container. So the container is kind of represent the foraging area, or like in general the environment for the ants can go around, but they still want like a nest. And usually ants prefer to stay in somewhere like it's dark and kind of humid and I would say a bit warm place. So I remember is in Andy's lab we use like some water, like those glass tubes, and then put some water in it, and then you suck like put a cotton balls into it. So it generates this kind of little water chambers. So first that space is narrow, and then the ants can also get waters from them. And then we also put some red color papers wrapping the tubes because the ants like dark, and then they cannot really see the red light. So putting red paper basically gives them a dark place. So you'll see like most of the ants will stay in the tubes where it's humid and dark, and you'll see the queens, which is usually bigger than the workers, so you can distinguish them from the workers. And you'll see they moving like the food around and moving the babies, which is called larvae, or the teenagers, the pupas around. And it was really fun to watch. And for me, it's like when I was doing research, I was focused on the workers, which are the majority of the ants. They're all females, but they don't reproduce. They only do the work for the colonies. Right. And we're interested in how they are behave because they are the most active one. The queens are usually just sitting in the deep of the colonies and reproducing babies, but not really moving around. So during the time I was feeding, I can see like other behaviors rather than just the snapping, because you saw the ants going around to get food or they're taking care of the babies. But sometimes, most of the time, I don't really know what they're doing. I can feel like there's some of the I would say behavioral structure going on in the colony, but I don't know what they are doing. And that actually raising more curiosities, or my interest into the ant study is, I think I'm more interested in this social behavior rather than those individual behaviors. Even I think I will study some of the most amazing individual behaviors in ants, but those social interactions caught my attention. However, during my undergrads, my first projects, like independent projects, was not focused on the social behavior. It's still on the individual level because the lab is focused on the individual behaviors during that time. So the ants I was starting with my independent project was called Giganteps destructor, which is some really wild-looking ants. So usually we thinking about ants, they're like tiny, they're small, and usually they will have, some of them have big eyes, but a lot of them are having tiny eyes because vision usually is not the major sensory for them. But the ants I will study, they are bigger. There are some panorines, which represent some of the bigger ants, like they usually have fewer individuals in the colonies, and most of them are more like, you say, each of the individuals are more powerful, like they can do a lot of work, rather than comparing to some other ants that we are thinking like with giant colonies, and sometimes they even have different body size, but each of the ants will have like a really specific work to do, and then they usually won't change in the work that much. Like for example, tiny ants may just go for foraging. They cannot be in like the soldiers to fight with some bigger enemies, that kind of stuff. Right. But panorines are more likely they can do multiple kind of jobs. So they are more like, I would say, less social kind of side of it, but I think it's also a kind of adaptation for their kind of lifestyle. So the Giganteps destructor I was studied, they have giant eyes, which means they really depends on the vision. So they can see things really clear, and they're actually really active hunters to hunt for other insects for the proteins resources. Wow. And they can move really, really fast. Like one thing is, with their really good visions, they can see the prey, and they jump between the branches, like they are originally from French Guiana in the forest. And another thing is they can jump from branch to branches, so it's a really fast move for them to catch up with their prey. So the question I was interested in is how do they jump? Like not many ants jump. Right. And some of the ants, like the Dajia I mentioned, the trap giants, they can jump backwards. It's majorly using this force from the snapping. But Giganteps actually using their hind legs, like jumping like a mantis. So what we are doing was we try to make the ants to jump in the lab, which is not that easy.
Yeah.
It's like the ants will jump, but they're going everywhere. They are going all the kind of direction. So first we want to capture their behaviors under the high-speed camera to see what exactly happens when they're jumping. What we did is we set up this little platform. So they're basically like a two-part platform, and there's a gap between it. So first the ants are jumping forwards, but the ants sometimes going onto the wall, sometimes they go backwards, all kind of thing. So we found is, okay, we can use like a little petri dish kind of following the ants when they're walking forward. So you have the petri dish kind of blocking their way to go back and also kind of pushing them to jump forward. So that's the way we figure out, okay, we can make them to jump to the direction we want so we can capture what's going on. And during the filming, we realized the ants will raise up their legs when they start jumping, but at the same time, they will also raise up their abdomen, which is basically we also call glaster, which is the bottom part of their bodies. Yeah. And we are curious, if this raising up abdomen is helping with certain things during the jumping. So our hypothesis was either it helps to generate more energy for them to jump further, or we can the abdomen was helping with balancing during the landing. And how we studied it, it was okay, we want to control this behavior of the abdomen. If we restrain the raising up behavior of the abdomen, what would happen? If it's helping with generating energy, maybe the ants cannot jumping as far.
Right.
Or if it's for the balance, maybe the ants will crash when they're landing. So they did this I found it kind of funny, but may not be funny for the ants, is we put some wood glue on the joints between the abdomen and the thorax of the ants, so they can now move freely. And then we let the ants to jump again, and we found is they cannot jump as far as they want anymore. So that was supporting the first hypothesis, and that's the major stories of my first independent projects, which I also use as for my undergrad thesis, and that's the kind of the first step for me to jump into the ants work. So that was the initial part why I get into the ant study, and also it was because of my observations of ants during the feeding period, making me realize, I really want to study the social interactions in ants. I want to know what is going on in the nest. And during the time, I also was reading a book called Ants at Work, which was written by Deborah Golden. It's a spoiler, but now I'm in her lab now. So that book was focused on her study of the red harvest ants in Arizona desert. And up to now, she has been studying those ants for 30 years. So in that book, I would think it was decades of studies of those ants. So she was basically focused on what are the ants are doing in the deserts. So the red harvest ants, based on the name, they eat seeds. So the foragers need to go out of the nest and then to collecting seeds as food resources for the colony. Yeah. And it's different from a solitary animals like fruit flies or rats. Yeah. They go out, the major motivation is being hungry, so they need to collect the food and then they eat it. But for the ants, it's kind of different. It's more like a social behavior. It's because you only have a few portion, a small portion of the ants as the foragers going out to gathering the food, no matter if it's a seed or it's insects, or some of the leafcutter ants will gather in the leaves. But they don't eat on site. They carry the food back, and then they will store it for the whole colonies. And also sometimes not everyone just go for the food to feed themselves. They will eat the food and then they keep the food in their social stomach, and then they are basically vomiting to each other to share the food in the colonies, which is also the same thing for honeybees. So I think the motivations of the foraging can be quite different. Like it's for the ants and honeybees, that was more like a long time planning rather than like the short time needs of the food to keep them from the starvation. But anyway, those ants going out into the forest, going into the desert every day to collecting seeds. And they use the seeds not only for the nutrition, but also as of water resources, because they are living in the desert, there are not many water resources. And the seeds have this high content of the phyte, and they can metabolize the phyte into the waters. So the seeds are really essential for them, because we know, like for the food, if you are starving, you can keep for several days, but if you're thirsty and you couldn't get water, you can die really quickly. And the ants going out for seeds to get water, but at the same time, going out also means losing water because it's desert. The sun has this heat and it's drying up the things. So for the ants, it's always a trade-off for getting the water versus you're getting the water by losing the waters on the way. And the ants do this foraging every day, and this foraging behavior is also social. So it's not like everyone always go out to getting food. They have some kind of recruiting process. And based on Deborah's book, based on their long-term study, they found out there are two different, I would say, subclass. Maybe it's not subclass, because subclass we usually mention is they also have some polymorphism, but in the harvest ants, they're actually looking the same. So it's like they do different kind of job.
It's like a behavioral caste, almost.
Yeah, so one behavioral class is called patroller, which indicating they're more on the doing the pioneer job, going out to find if there are enough food resources outside or where the food are. But they are just a small bunch of the ants. They go out early in the morning, and then they patrol for a little bit and come back, and somehow they can pass the information to the majority group, which is the foragers, about if today is a good day to forage or which direction that in general we need to go or whatever, that we don't really know what kind of information they gather and then they pass. But patrollers' behavior does do affecting on the foraging behaviors later for the foragers. So after the period of the patroller, the foragers coming out of the nest. And also I want to mention is the nests are on the floor of the desert, so they're basically making those holes into the ground so they cannot get like too hot or too dry because the temperature and humidity are relatively stable underground. And then the forager get information, they come out, and how many foragers can come out for foraging is also based on some social information. So what they found from Deborah's work, it is based on the social interactions with their nestmates coming back with food. So the foragers that is planning to going out, they will wait at the entrance, they will have a chamber near to the entrance, and that was like an entrance chamber. They were waiting at the entrance chamber, and then the ants are majorly based, those ants are majorly using the information of olfactory information rather than the vision information.
Pheromones.
So they use the antennas to touch each other and to sense like the olfactory or, as you said, the pheromone information. So when they're waiting there, they will going to interact with the ants with food coming back. And it's based on the frequency they sense this individual is my nestmate, and also they come back with the smell of the seeds. At a certain frequency, it kind of reach to a threshold that tell them that, okay, it's a good time to go for foraging. So that's also giving the information is if there are more nestmates coming back with the food, the more likely the other foragers will come out to get food, which indicates because there are plenty of food outside, so we need more workers to help with foraging. So that generates this kind of positive loop to really focus on getting the food when the food is abundant. And Deborah did this really interesting behavioral test. Like that's the way they figure out how this recruitment happens. Is they recording what's going on in this entrance chamber, and then they coated some glass beads with the smell of the ants with the seed smell.
Oh, clever.
Yeah, and then they just drop the beads into the chambers, given to the incoming foragers. And they found out is when they give the beads at a certain frequency, then it's the foragers are more likely to come out for foraging. And for me, when I was undergrad, I was like, wow, this is such a cool work. Like, because I can imagine it would be so difficult to do behavioral tests like in the field. Like when I was doing the jumping ants, it was in the lab, it was highly controlled. It is still really difficult to make the individuals to do the things you want. How do they do that like in the field?
With a complex social like colony.
Yeah. So that was like, oh, I really want to see how they do it. Like I want to experience that, like to see how this work. And I was really fascinated by this idea that described in Deborah's book about the collective system. So she called the ants colony as a collective system, which means this system is now central controlled, because even the ant has queens. The queen will not tell everyone what you need to do, because they are inside of the nest, like, and giving birth to the new ants. But she cannot tell like who is going out for foraging, or it's time for everyone to stop foraging, or there are certain work like cleaning up the colonies or taking care of the baby ants, like who need to do what. This is all depending on some information that is based on really basic interactions between the individuals. But for me, it was really fascinating, like how this kind of, I would say, high level structures of the colonies, or like how to make the colonies to be functional at this high level, just based on some really basic interactions at like one ant to the other ant's base. I mean, it's difficult to study as it sounds, but that question for me is more interesting than some basic behaviors, or like why I'm more interested in ants over other animal systems. Is because I'm really into this collective behaviors. And also I was like, yeah, this is really challenging, but at the same time, I really want to know how this works. So that is how I get into it. It's kind of a long answer for this whole thing, but yeah, that's what's happening.
Yeah. I mean, that's why I think ants are so interesting to me, is because of the way that they cooperate and work together is, I mean, mind-blowing to me. And, you know, how they have such specialization for certain tasks is so cool. So tell us a little bit about where you've like taken that. What was your next project like?
Okay. So after I graduated from Illinois, which I can give a timeline I think I should give earlier. So I came to the U.S. in 2015, and I graduated in 2017.
That's cool.
So during that time, I was like, oh, I got experience in the ants lab, but really it's just really focused on the individual behavior. But my interest was driven to, it was twist to the social behavior. So I was thinking maybe I can get some idea of how people study the social behavior of the ants before I decide if I want to go to a PhD program to focus on this project. So in 2017, I moved to Boston, and I joined 2 labs at the same time as a research assistant. It was kind of funny. So I got my first job in James Chanello's lab in Boston University, who is really famous for his study in the social brain hypothesis in ants. So I can give you some information about this social brain hypothesis. This was started with some anthropology studies in primates and humans. And the hypothesis is saying the size of the brains is correlated with the social levels of the animals. So in the primates, the idea is the animals that is more social or have a bigger social groups usually will have a bigger brains because there are certain functions for social behaviors or learning behavior and the communications are important that you need a bigger portion of your brains to do those works. So I think it's kind of straightforward that for some smaller monkeys, they have smaller brains, while for like chimpanzees will have bigger brains. And this is talking about like relative to their body size rather than the absolute size, although those do have like a smaller monkeys have smaller body size, but the brain proportion is lower compared to a chimpanzee. And for human is one of the animals that has the biggest brain compared to all kind of animals. And that's why humans are more social than most of the animals. That's kind of some classic theories. And then some people may be interested is how about the insects? Is that also work for insects? So the lab in Boston, they're focused on to compare the relative brain size in different species of the ants, and also about the energy using of the brains in these ants. So the story can be much more complicated because we know some of the ants have the biggest colonies there. Each individual ants are tiny. And based on the measurements, it's not necessary that the ants with the biggest social group will have the biggest brains, which is also kind of makes sense because previously when I talk about the paneral ants, the jumping ants, they actually each individuals are capable to do more work, like all kind of works compared to like, for example, leaf cutter ants. Although the whole colony is so complicated, like there are like 4 or 5 subclasses and then focus on different jobs. But if you only take one ant, they are just focused on one kind of work or one aspect of the work. Like you cannot make a nurse to be the soldier at the same time. So this series need to be like explained more or improved in the ants' words, or you say the insects' words, because the social complicity is not also just based on the social, like the size of the social group. It's also based on, for example, the polymorphism, like how complicated of this division of labors happening in the group. There definitely can be one factor is how big the colony is, but also there are also part about the social structures in the colonies. So one thing that James Labbert's study is, although the brain size is not correlated with the social complicity, but for the colonies that have like a different kind of like polymorphism or different kind of labor specialties, the ants will have bigger proportion of certain part of the brains. It's bigger compared to like other colonies have like a smaller social group. For example, one part of the brain is called, or like I said, the brain parts. One of the brain parts is called mushroom bodies, which is some of the, if you see an ant's brain, you will see it's like basically a mushroom shape of the part of the brains, which is really important for neurons that is controlling learning behaviors and communications. So it's a center of the social behaviors. So we found, in that lab, we found is the colonies that is having really complex social dynamics and social division of labors. The ants, even they have a smaller brain, but the mushroom body will occupy most part of the brain compared to other colonies.
Okay.
And another thing they found is the usage of energy of the brain is compared to all the body metabolism. The metabolism of the brains is the highest. So you can see the brain is really costly compared to other part of the body. So even you have a relatively smaller brain, your brain is doing a lot of work. It actually take advantage of using a lot of energies to make sure the ants can accomplish certain kind of social behaviors. Yeah. So in that lab, the major work was dissecting a lot of ant brain from different species.
Wow.
And that was the place where I learned how to dissect the brain, like a tiny insect's brain.
Wow.
And so I was helping with a PhD student during that time to dissect the brains, and then we also measure their metabolic rates of the brains in a little fancy machines. And I was also working on relatively independent projects about like how the nutrition is affecting on the brain energy usings. It's basically we use the pavement ants, which is a really common ants that you can find anywhere, especially in Boston. So we collect those ants, and then in different colonies, we give them either high protein food or high sugar content food or just waters. And then we look at, like, we feed them for sometimes, and then we measure different kind of physiology or activity, trying to figure out if different proportion of the food, if high proteins or high sugars have effects on, for example, the survival of the colonies or the aggression behaviors, or the metabolism of the brains. So this work is still in the process that we are trying to wrap it up, even after I already graduated, but usually publication takes some time. So we are still in the process of analyzing those data, and then first we know it's obvious is starving the ants definitely causing them to die faster. And the protein food is important for the ants' development because for the larvae become the adults, they definitely need to use a lot of protein as the resources to build up their bodies and stuff. But a high protein diet usually is also toxic. So like a relatively lower level, lower portion of the protein diet will be helpful to help the colonies to grow, because you need the babies become the adults. But definitely too much proteins can causing a rapid, drastic dying of the colonies for sure. And we are also looking at the aggression behavior. Basically, we just take the ants from different diets group and put them together to see if they show any kind of aggression behavior, like if they'll fight or have some social interactions. But we didn't really find much of the difference in aggression behavior. But we also found is even with just water diets, basically by starving the ants, it will affecting on their behaviors, because they will get slower and less active. But when we measure the metabolic rates of their brains, they're not dropping that much. So that means also kind of supporting previously when we talk about the social brain hypothesis, that the brain is really costly and they are kind of the priority for the function. So when you don't have enough energy, there's still a priority to distribute the energies for the brains to be functional. That is what we found in that project. But at the same time, when I was working in Boston University, doing all these social relevant projects exactly what I want to study, I also got the offer from a lab in Harvard. It was kind of funny. So I sent out the job application in February 2018 or something, and then they didn't reply to me for 4 months until end of May. The PI suddenly told me, "Oh, we got some money. We can hire a research assistant. Do you want to help with this project?" So I said, awesome. "Yeah, I can do that because I'm in Boston." So it was funny. Half of the week I would work in Harvard, and half of the week I can work with the ants in Boston University. And in Harvard, my work is less relevant to the ants. So that lab is Naomi Pierce's lab, who is focused on butterflies and ants. I think for her PhD project, she was focused on the mutualism behaviors between ants with caterpillars because it's basically there's some species of the ants would provide basically living shelter for the caterpillars. They will take the caterpillars into their nest, and that way caterpillars can avoid like predations or any kind of dangers from the outside world. And ants also kind of feeding caterpillars because they get food and then they share in the colony. They can also give that to the caterpillars.
Yeah.
And in return, the caterpillars release this kind of sweet creations. And the ants love it. So they use the caterpillar kind of like a cow.
It sounds a lot like the mealybugs that some of the ants farm. I had another grad student on a couple weeks ago, and that's what he studies. So it reminds me a lot about that. So I didn't know they did that with caterpillars too. That's really
cool. They
cool.
also do that with butterfly caterpillars. Yeah, and then but that was the PI's previous project. So that's why the lab have these two directions. Like one is focused on butterflies, and the second one is the ants. So they hired me majorly is to help with one is the ant feeding, and secondly, there are like postdocs that working on like evolutions of butterflies projects, and they need research assistant to help with some molecular work like PCR and stuff.
Yeah.
So my major job is not really focused on ants, but help with the PCRs, but that was the chance for me to starting to learn about bench work. And besides the bench work, the feeding ants, there's also jobs in the Harvard Natural History Museum in the entomology collections. So that professor I'm working with, she's also the curator for the Lepidoptera section. So during that time, they are trying to digitalize the records of their collections of butterflies and moths into computers. So my job was including going through some really old like loaner sheets that was like handwriting or like by the typewriters, and then trying to figure out if they returned the specimen and putting the informations into the computer. So people don't need to rely on those really old papers. Mm-hmm. And that was also the time I get the chance to see those specimens in the collections. Like that was the first time I worked in a museum. I didn't realize how big the backstage of the museum could be. Like when you go to a museum, it's like, oh, there's a lot of things, but actually there are like much more, like 10 times of the things in the back.
Basement, yeah.
And yeah, it was really fun to see all kind of those Lepidoptera specimens. And there was also a PhD student during that time was doing some research on the sex dimorphisms in the butterflies' wings, not only by the colors that we can see, but also the colors that the butterflies can see. That's because, so we have those proteins that is for the different color sensory called opsin. And I think we have 3 different opsins that is really focused on like sensitive for certain wavelengths of the light.
Yeah.
And that's causing the combination of all the colors that we can see in our life. But different animals will have different numbers or different types of opsins, which can cause their vision can be really different from us. And for butterflies, they have 4 rather than 3 opsins. So that causing them can see the wavelengths that we couldn't even see. So some butterflies you see, they look similar, but actually for the butterflies, they look really different. Mm. So I remember they're using some special cameras with can see the ultraviolet or ultra red, one side of the waveform that we couldn't see.
Yeah.
So they can capture the patterns using the cameras like when they are shooting on the butterfly wings, so they can see, so that could be how the butterflies see those patterns. So even we cannot tell like the male or the females based on the pattern of the butterflies, but the butterfly themselves can see it. So that was also part of it. So it's really interesting because that's the way I can help them to prepare the specimens they want to take the photos. like they already have really beautiful colors, but then they were using these cameras to shooting the photos, and then you will see, although I remember it was less fascinating as they don't really show like the crazy colors, but they are black and white, but they are showing difference in the patterns because of the shades of the black and white can be different.
Yeah.
But that was still kind of surprising for me when I was working there because I never really think about those kind of questions.
Yeah.
But still, I'm an ant person. I'm still really into ants. So sometimes I ask my PI there about, can I work in the ants collections? She's like, I'm not in charge of the ants collections, but there is an ants room in the museum. And I've never been there. Like she said, none of my lab members have been there before because we don't work with them. But there's an office across the way of the ant room, which previously when I went to work, I passed there and I saw it. It's E.O. Wilson's office.
Wow.
And E.O. Wilson is like a kind of really big name and like kind of the father of the ant studies. During my time when I was there, I don't think I really see him. I think he already retired. Although I do hear people he still come to campus and go to the office sometime. But it was quite exciting when I saw that office.
That's cool.
And it was also exciting to know that because before he was retired, he was also working in the ant room. So my PI, Naomi, told me was, okay, so if you get a chance, maybe you can go to say hi for the curators in the ant room. It was Stefan, I think. I think he already left during this time, but previously he was there. And the first time I went there, after my work, I went there to see him, and he was like, "Oh, I have been waiting for you for a long time." It was really funny. It was because at first I want to be polite, so I sent him email saying I'm interested in ants and I want to see the collection. Also, I'm interested to study, to learn about the ant taxonomies from him. And I sent like 2 or 3 emails, he never respond. But when I show up in the room, he's like, I'm waiting for you. So how could I know that, right? But that's how it works. So I went there for like every Friday afternoon just to sit there, and then he will give me several species of the ant specimen, and then he will give me a guidebook and saying like, just this, figure out what are those under the microscope. So it will be just a whole afternoon. I sit there putting like 10 different specimens one by one on it, and based on the guidebook trying to figure out what genus or the species of the ants is, and I write it down on a notebook and give it to him, and he will tell me which one is right, which one is wrong.
Yeah.
So that went for several months, and even up to now, I still really precious those times in the ant room, like doing nothing but just looking at the ants. But anyway, that was a whole year for me before I decided to go for my PhD projects, that I feel like I'm ready to go to an ants lab to study social behaviors as my PhD projects.
Yeah. So tell us about that. What was your PhD in? Because you just finished it a year or two ago, right?
Yeah, half a year
Half
ago.
a year?
Actually.
Okay, yeah.
Yeah, so it was 2018. I moved to Philadelphia and joined University of Pennsylvania. During that time, there was an ant professor he studied focused on the evolution of social complicity in ants. So I was like, yeah, that would be the perfect match.
Yeah.
And I was planning for my PhD thesis during my first 2 years as the graduate student. My project was focused on the Trophallaxis in ants. So the Trophallaxis is that process that I previously mentioned. Is the foragers get the food, the ants, the foragers may eat the food, and then they're going to share this liquid from their social crops to like other nest members or the baby ants or the queens.
Yeah.
So the system I was using is the pharaoh ants, which is a tiny ants that it's really frequently found in like human living buildings like hospitals or apartment
Yeah.
buildings. So those ants are really adapted to human environment. Like they don't need really big space for their
Okay.
nest. Like in the lab, we're usually just using 2 slices of glasses and put some little space between the glasses. But the ants love those narrow space.
Yeah.
And then that's the way it's easier also for us to observing what's going on in the nest because it's glass, it's transparent.
So they'll nest like between like floorboards or like in the walls of buildings then, I would imagine.
Yeah, it would be like just a tiny hole or something, like a between like a gaps between the walls.
Interesting.
Yeah, and then those ants are also easy to rear in the lab. That they don't even do mating flight, which is the kind of courtship seasons for the ants. Most of the ants will have this kind of courtship rituals. So usually when you see, that's some of the only way you can see the male ants and the queen ants going out, and those ants will have this wings so they can fly in the airs. It's based on different species, but some of the species will do that in the early spring. If you see there are some flying insects outside, but when you catch them, actually are ants with wings. That's what's going on. So during the mating flight, the female will get fertilized, fertilized, and then they will store the sperms from the males, and then some of the species that living either on the underground or on the woodlot, then the queens will shade out their wings and then starting to dig into the surface of where they want to have the nest, and then give birth to the baby ants.
Right.
And that's how a colony is generated. But for the pharaoh ants, they don't even need to do this. Like the males are just going to meet with females like in the nest, so sometimes it's causing inbreeding. But I think in the wild it's less likely to happen, but they don't need to fly around, they're just crawling on the floor and surface and stuff. But what I was interested in is, so the nurse, we say the ants that feeding the babies, we call them the nurse,
Yeah.
and then they eat the food, and then they release this liquid to the babies, which should including the nutrition that is important for the baby to grow up, and also there could be some chemicals the nurse release in their social crops, and then passing that to the babies. And when they're passing the food, we call this process as trophallaxis.
Yeah.
So my question is how this trophallaxis affecting on the development of the babies. So previous study in this lab have been shown there are some nurse, even in the nurse, there are division of labors happening, because if you're just tracing single nurse, that when they got food and then they go to different babies, we found is some of the nurse only focus on feeding the early age babies, like smaller babies, and some of the nurse only feed the older babies. But also there are some nurse, they don't mind, they feed everyone.
Weird.
So we are wondering for the nurse that is specialized feeding the younger babies, if the nutrition, like the liquids, contains different kind of compounds that is different from the nurse that specialized feeding the older babies. So to study this, it would be difficult because you need to first tracing different individuals.
Yeah.
For this behavioral tracing I have done, it takes sometimes 2 hours to just looking under microscope in a colony of the tiny ants, and then you say, okay, I'm just going to tracing you, and you see the ants going for different babies to feed them, but sometimes they feed 2 babies and then they're done today, they do other things.
Wow.
Or they decide not to feed and then say, okay, you are wasting 10 minutes for me, I need to move to another individual.
So, did you ever paint certain individuals so you could track them easier? Or was it literally just by eye?
So for this part, we only do by eye because the goal is to collecting the nerves that we know if they're specialized or generalist.
Wow.
If you print them, that is actually one of the postdoc was working in the lab was doing. The thing is, the painting for some species it works, but you need some time for the ants to recover after your manipulation because a human modification of the ant by any means can seriously affecting on their behaviors.
Yeah.
Like they will not doing what they're supposed to do for some time. So usually what you do is you paint them and then you put them back to the colonies, and then maybe next day you come back to observing the behavior.
Yeah,
so this
that
first
makes
will take
sense.
some time. And secondly, the postdoc did succeed in painting some other ants in his PhD work, but he was trying in the pharaoh ants, and it kind of didn't really work really well. The first is the pharaoh ants is so tiny that it's more technically difficult to put their pins on it. And secondly, those ants have this behavior that they really like grooming each other.
Right.
So the case is he put the pins on, and the second day the pins disappear because the ants just bite it out from each other. So it doesn't really work that well. And for the projects I was doing, tracing the behavior, it's kind of rough. Like every day, just looking under the microscope for like 2 or 4
Yeah.
hours, but it's still possible. It's like to trace one individual, and then it's like my goal was to observing the ants if they are going to feed like maybe 5 different babies, and if they're all like like a certain age period, like a young age,
Right.
then identify this one is the specialized in like a young baby feeding. Then we use a forceps just to grab it out and put it in a little tube for my next step. So that's the way we can collecting samples. But this is for like some pretest that we want to do is to collecting the different different nerves that is specialized for different age, or they are generalist, and then we want to get their brain and compare the gene expressions in the brains to see if any genes that is relevant to the metabolism or the foraging could be different, that causing them to do different kind of nursing behaviors. But another part is we actually are more interested in what is in the social liquids they are passing around. So we want to collecting those social fluids. So that is the part I didn't even get a chance to do it, but it was planning to do it, and it's super difficult. So the case is you're tracing the ants and identify them for a certain kind of job, and then you want to use a super long pipette to collecting the liquid when they are feeding the babies.
Wow. Yeah.
So we are trying that. It was difficult
Yeah.
because sometimes you lose the track of the ants for the behavioral part, and secondly is how do you put the pipette on site to get the liquids? What if the ants doesn't like it and then they just move away? Or it would be super hard to get the amount of the liquids that actually we can do any analysis on
Right.
it. And there is other work in not pharaoh ants but carpenter ants, which is a much bigger ants,
Yeah.
that they succeed to get some liquids out, and a way they are doing is to squeeze their abdomens to force them to warm it to get the liquids. So that was also a strategy we're considering, although pharaoh ants are much, much smaller. But before I was trying to figure out how to do this, a kind of a dramatic event happens in my life. It was 2020, I think. It was like during my end of my second year that I was proposing to do this project. And my PI during that time has been moving to Texas because his family is there, and it was pandemic, so
Right.
he was with his family.
Yeah.
And there was one day just actually like 1 week after my qualified exam, which is the exams that as a PhD you need to propose for your thesis project, and then there are several professors, including your PI, make approval that you can do this project for your PhD. So I finally get approval saying, okay, this sounds really tricky project, but you are allowed to do it. And I was really happy for that. And then my PI video chat with me later on saying, oh, so I decided that I'm going to move my lab to Texas, that I'm not going back to Philly anymore.
Wow.
So you can decide if you still want to be my graduate student, and you can also move with me to Texas, or you can decide to stay, but then you need to figure out where you are going. So that was a really tough decision for me. I think I was struggling for a month, and I talked to different people to figure out what are the better choice. So I feel like I still want to study ants. Even if I change my PhD projects, I still want a way that I can go back to the ant study. So in that case, maybe moving to Texas would be a good idea, that I can continue my projects, but it is also a lot of uncertainty going there, because it's a place that I've never been before.
In the middle of a pandemic, too.
Yeah, that was the tricky part. It was like moving during the pandemic across the country. And also, when the PI was there, the ants are still in Philly. So if I'm going to there, we also need to figure out a plan how to move those ants to Texas. And because it's kind of rebuilding a lab in a different place, that it may take longer time for me, because I don't think I can do anything until the lab is being rebuilt, and also during a pandemic, so who knows. So in the end, I decided I'm now going to move with the ants to Texas, but I'm going to stay in Philadelphia to finish my PhD, and we will see how it goes. So during that time, there is a lab that I'm kind of interested, and then the PI is also recruiting new students. It was actually the lab I did my PhD thesis. It was Yunding's lab, who is a new PI during that time. I'm basically her first graduate student. And this lab is studying things that's totally different from the ants. So their focus is the courtship behaviors in fruit flies. So I don't know if you ever know that the fruit flies sing courtship sounds like crickets.
Really?
So they vibrate their wings. The males were chasing the females, and then they sing courtship sounds by vibrating the wings to the females.
No way.
So we don't even know if how the female choice, if they are like the birds, they judge the singing of the males to decide if they want to go with them or not. But because there are not much work have been done on this part, but definitely the singing behavior is really critical in the courtship process for the fruit flies. And fruit flies is really different from ants. First, they kind of live in a group, but they don't have this really complicated social structures. And they are kind of model species. So in the ants, most of the work are really focused on ecology or behavioral aspects. There are some species that people can do genetic modification, but it's really hard, and it's really based on special ants. Like, for example, one of the ants that can do genetic modification is because those ants, they can reproduce by cloning themselves. So it's not only queen can reproduce, it's all the workers can reproduce. And they are basically making the identical copy of themselves. So it's like a reproducing of cells rather than like a typical ant colony. But it's different from fruit flies. So fruit flies is like a lot of genetic work and neural work have been done in fruit flies, and it's for how many years, hundreds of years, people are working on that. So they have tons of tools to do genetic modifications, labeling specific neurons, and then they can even modify the neurons' activities to see how this is affecting our behaviors. So I would say if ants and fruit flies are two maps, it's totally in different scales. In the ants map, it is really fun to see, but there are not many details. And it's really hard for people to figure out exactly what are the details on those maps. Well, for the fruit flies, you get this really old but fully details of the navigations, where to go and what are the part of different area, that kind of maps.
Yeah.
So it's really different scenario. So for me, it was really challenging to join and work in a fruit fly lab because I was working with this kind of empty maps for a long time, and then I need to work on this full detail maps that people have different kind of mindset when they're looking at the questions. People will have different way to work in the labs, because in ants, major work is like a behavioral trackings, or making the ants to do certain things and see how it, like, or you modify certain things in their environment and see how it affecting on the behavior. Well, for the fruit flies, you need to generate transgene flies that you'll really focus on. At certain time, you need to collecting the offsprings of the fruit flies, or they call virgin, which is the female flies just coming out without having mating with any males.
Yeah.
And then you want those virgin females to mate with certain genotypes of the males to generating a certain kind of genetic combinations of the offsprings, that kind of, so the timing is really critical. So that affecting on how I was working in the lab, like I have to be there at certain time to collecting certain kind of flies, so I can make this crossing to generate the next generations that I want for my experiments. Right. And also there's a lot of brain dissections, because I was working on the neural mechanisms of the courtship evolutions in several species of the fly. So I used the model species Madagascar, but there are also some sister species of Madagascar. And the big question is, in the courtships, some of the fruit flies will have different kind of sound types, which is basically I'll say like a different components in their sound. So I can kind of do a really bad mimics of the fruit fly singing. So Madagascar have these two types of sounds. One is called sign sound, which is kind of a humming sound. So it's like a lower volume, and it's kind of continuous sound, like a ooh sound. And then they also have a second sound type called pulse sound, which is more like a drumming sound, like they have a more intervals and it's like a higher volume. So instead of a ooh sound, they sound like a do 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 do. So you can hear those gaps between the notes.
Interesting.
Well, yeah. So in the a sister species called Yakuba, this species, they don't have the humming sound, they don't have the sign sound, but they still have the pulse sound. So all the sounds they sing is having those intervals. And the question is, why these two species sing different kind of songs? So because the Madagascar is the model species, people have done so much work in it. They found out there's a neuron in the central nerve system, the neural central nerve systems of the flies is really important for generating this kind of songs. So if you inactivate this group of neurons, then the Madagascar can only sing pulse sound. They don't sing sign sounds anymore. So it's more like a Yakuba. So our question is, how is this group of neurons looks like in Yakuba? And if they're different, maybe the difference in the neurons if results in these behavioral variations in what kind of sounds you can sing. So my PhD project was majorly just focused on this group of neurons, and I compared this group of neurons in different species, like a sister species of Madagascar, and see if I manipulate these neurons, how this also affects on the behavior. So if I just telling the story of the Yakuba, the one lost the sign sound, what we found is this group of neurons still exists in Yakuba, but they have much fewer numbers of the neurons compared to Madagascar. So certain types of this group of neurons has been lost. And that could be the reason why they lost this sounds, because with fewer numbers of these neurons, they are failed to control, like to activate the downstream neurons to do the work. And another thing we did is we found there's the certain genes that express in those neurons, and also is important for those neurons developing into the way that they are important for generating the sounds. In Yakuba, those genes express there, but actually those genes causing the loss of numbers of these neurons during the development. So if we keep those genes to express, which kind of take out the functions of those neurons to be take out during the development, so you get more neurons growing out if you don't express those genes in Yakuba. And that actually can also causing the moving the wings in a certain way that indicates they can rescue the behavior of singing, although they didn't sing the sign sounds, but they actually starting to wave the wings.
Wow, that's
so it's a really fine detail of study, like how this behavior is generated, why there are different species, and then we can track down into a certain neurons that if because of these neurons, the behaviors are different. And why these neurons are different to different species is because this gene has these special functions that they keep the neurons growing in Madagascar, but causing neuron loss in Yakuba, and then causing this behavioral diversity. So for me, this is amazing that I cannot see anything I can do at this level in the ants at this point. But at the same time, it is such a different mindset, like when you look at the behavior in fruit flies and you're thinking about the genes, the neurons, and also you can find the actual tools that to test on your hypothesis. Well, in ants, it is so hard that you trace back to a single gene or something to figure out if that affects on the behavior.
Yeah.
So for me, it is a really precious opportunity to learn how this is doing in the fruit flies, and then definitely now like a postdoc kind of period of work, I can make it work, but in a long term, I was really hoping I can do something that can make some of the neuron or genetics tools being available for the ant study, because there are questions that is really cool that we can do in the fruit flies, but there are also the behavior, especially those social behaviors. The fruit flies just don't do those. So you cannot study this question using the fruit flies. You have to study this in ants.
Yeah.
So I want to find if there's a way to do it.
Yeah. So you've got kind of a dilemma, because fruit flies are such a great model system. We know so much about fruit flies, and there's so much data out there and tools out there that you can use. But they don't have the social structure that ants do. So it's like you can't really use both as like each has their own purpose, right? So I actually wanted to ask you about what you're doing now at Stanford. So you're in Dr. Gordon's lab, and that's a crazy full circle moment. What was that like to get to work with her, and like how has that experience been so far, and what are you working on?
So I think it was one of my happiest moments when I got the offer from Deborah. It's like I saw she is recruiting postdoc when I was about to finish my PhD, and I was like, oh, that would be such a good opportunity that it feels like a dream could come true if I can work with her. But at the same time, it's like she is like a really she is famous for doing the field work, the ecology part of the work. While my background from the fruit flies are more on the neuron and genetics background, I don't really know how this combine to each other. So but I was like, I really like her work. I would be regret if I'm not applying for her lab, and she's recruiting, so I'm going to talk to her anyway. So I talked to her, and she told me, oh, actually, I am interested in the neural aspect of those ants. So if I just give you a quick rewind about her work, is she worked she works in the foraging strategies in the harvest ants.
That's right.
That they go out to get the seeds and getting the waters from the food, but at the same time losing waters on the way for foraging. So the previous study from her lab, like by a graduate student did, is they found out some colonies will have different kind of foraging strategies when the temperature is really high and the humidity is really low. So that is going on with the global warming, that in recent years, like the local temperature and humidity just going crazy. There are some of the days in the summer just so hot and dry that like a human don't really want to be there
Right.
at that time. And so is the ants. Like some of the colonies will just stop foraging on those really hot and dry days. But some of the colonies will still go out, and no matter how hot or dry it is, they're just consistently sending foragings out to get food. So in regular days, like you would say, going out for foraging is always a good strategy, that you just get more food back. But on a really dry and hot days, there could be less food abundance outside of the nest, and also there's higher risk that you lose water or even die on the way for foraging to look for much fewer seeds. So there is a kind of a trade-off, or like a decision for the colonies need to make, is when the temperature is so extreme, then should they still go out or not? And definitely ants don't do like a group meeting or something to say about this before they go out.
Yeah.
It's still kind of based on those individual social interactions at the entrance lobby, figure out if there are more individuals coming back with food or not. And the previous graduate students want to link this part of social interactions with this decision makings in if they're going out for foraging or not. And what he found is dopamine, which is a really important neuromodulators, can play a role in this. So in other studies, like in humans or vertebrates or even fruit flies, that we know dopamine is important for decision makings, for going for higher reward, or if you go for a reward with a risk of like losing the reward, or you're going for a more certain but smaller reward, that kind of stuff. So dopamine in general can promoting the motivations to go for bigger rewards, that with the increased dopamine levels, that the animals are more likely to overcome their aversions or like their scares of risk to go for like a reward. Usually it's a forage, like a food resource, like a food reward.
Right.
So in the ants, could be similar things going on, that maybe the dry and hot environmental effects is a big stress, or say a high risk for the ants, that can affect on the decisions to going out or not. But maybe a high dopamine levels in their brains can cause them to be more motivated to go out, even it's really dry or hot. But we don't know exactly what happens. So based on previous research, what we know is if you feed the ants with dopamine, like 1 day before, and then you send them back, and the second day, the ants are more likely to come out for foraging, even if it's really, really dry and hot. So dopamine, a high level of dopamine definitely can cause them more likely to coming out for foraging. But we don't know how exactly that is work for the colonies that decided to stay, or the colonies that always going out. Like if there's a dopamine levels changes, variations in nature in both of the colonies, or is the way of dopamine regulating this behavior is different in those colonies. And no one has ever been looking into the brains of those ants yet. So that's how I jumped into the scenario. It's like, oh, that would be perfect,
Yeah.
because I do have the background in the neuron. I I can figure out how to visualize the neurons that releasing dopamines. And at the same time, I'm really interested in this social behavior. So I want to use these neuron tools I learned from fruit flies to study these questions in the harvest ants. So during the summer, I went to the field with Deborah. That was the first time I went to her field. I was reading in the book 8 years ago or something. So we saw those ant colonies. Like Deborah knows each of the colonies. She gave them different names, and she knows which colonies the queen was originally the daughter of that colonies. So it's like a grandmother to mother to daughter, that kind of thing, which is really crazy. Wow. And she also knows, based on their long-time tracking of the behavioral data, that some of the colonies always reduce foragings during the hot days, and she called them reducer colonies. And some of the colonies always keep going on with foraging, so we call them non-reducer colonies. So she have all the records of that. So during my field work, my work is every morning I go out, get the data about the temperature and humidity during the time we are in the field, and then I will go for the colonies that, based on the behavioral records for previous several years, that the colonies definitely are reducers or non-reducers, and I wait for them to start foraging and to collect the individuals that coming out as foragers, which is, you can see as on a regular days, that would be easy for both of kind of colonies, but on the really hot days, it is really difficult to collecting reducers because they don't want to come out.
Right.
Yeah, so sometimes we just cannot get the individuals, or it will take a really long time to wait them to come out, or it's just a few of them come out, but I need to collect them. And the reason why I need to collect them is I want to measure dopamine levels in their brains under certain kind of environmental conditions. So that way we can measure their dopamine levels in both of the reducer and non-reducer colonies, and then across different kind of temperature and humidities. And that's the way we can try to figure out first. It could be the case that non-reducer just have higher dopamine levels in general no matter what. Or maybe it could be the dopamine levels is higher in the non-reducers on the hot days, but lower in the reducers on the hot days, but maybe there's not much difference on the humid days. And based on our preliminary data, it's more likely the second case.
Wow.
So we saw is the dopamine level is kind of stable in the reducers, actually. They are kind of similar on hot days and the dry days and the wet days. But for the non-reducers, we saw it's much higher of dopamine levels when it's really dry, but it dropped to a relatively lower level when it's wet. So that gave us some rough idea about how dopamine changes in those colonies. So first we know there are differences in their dopamine regulations in different colonies, and somehow the behavior, the dopamine level is higher in non-reducers, which makes sense. Then they can overcome the risk of losing waters on the way for foraging. So that's why they're going out.
Wow, that blows my mind. And when you were talking about the certain ants that will go out during that high risk, very hot day, it almost makes me think of like a thrill seeker, like someone who, you do a risky thing like skydiving, right?
Yeah.
It almost reminds me of someone who is a big thrill seeker. So you go on, you go skydiving, and you have this big rush of dopamine, right?
Yeah.
That reminds me of that a lot. Like these ants go out, do this thrilling thing, which is going out during a high risk time, and their dopamine levels change according to that. That's crazy to think about and really cool.
And also it's kind of just we got this picture of what's going on like in the middle of the thing. Like we don't know why they will increase this dopamine.
Yeah.
How do they sense that? Like do they sense the humidity and then that's what affecting on the dopamine releasing, or what kind of mechanism causing it to get higher, and then how this higher dopamine affecting on this foraging decision process. Like we're just know what happens in the middle. Like we know the dopamine change, but we don't know why it's changing. We don't know how this changing impacting on the behavior. But this is like a fascinating questions that I want to see how to study them. Yeah.
I'm curious, are you going to look at all at if there's like a genetic aspect? Like, are these, what did you say they were called? The certain colonies that don't go out as much as others.
Yeah, they're reducers and
That's
non-reducers.
right.
Yeah.
So I was wondering if you're going to look at all at if there's a genetic aspect to a colony being a reducer versus a non-reducer. Like,
Yeah.
do colonies that are non-reducers give rise to more colonies that are non-reducers? Like, is there any genetic aspect to it?
Yeah, that's a really good question. And Deborah actually has some insight on this. So first, because of the behavioral results, the behavioral data she had for like 10 years or 20 years, there is consistency of the colonies that if this year they're non-reducers, they always are non-reducers.
No way.
So for the colony structure, that the workers only live for a year. So a second year, the workers are totally different group of the ants, but they are from the same mother because the queen can live for decades. So it's from the same mother. The workers are all have the same strategy. So that's the evidence showing there could be a genetic components going on. This is an inheritable trait.
That's crazy.
And secondly, Deborah is also doing this work for basically doing like a genetic tags to figure out the lineage of the different colonies in the field. And her current results showing there are two major lineage in the field that even they have like hundreds of colonies, there will be a majority of them are from one lineage, and then there's also still a few of the colonies are from a separate lineage. And even there are a few, they're kind of stable. They're always there. They're never being overtaken by the bigger lineage. And we can even see the difference from the lineage from their behavior. For example, the way they build their nest. Like all the nests are underground, but they will always have these holes, like indicating the ants were coming out. It's their entrance or the exit. For the one lineage, the nest is always kind of simplified. It's just a hole on the ground. And I mean, it's a desert. There's some grass growing around it, but usually it's just a bare desert, like a soil, and then you see a hole over there. But then the other lineage, I think it's actually the minority lineage. They have a really like a dramatic nest. Is they will put little stones around the nest. Like you'll see is the sediments of the nest are different. Like you walk on the desert and then you saw there's a pile of tiny rocks accumulated around. It looks like a micro habitat.
No way.
And then you see there's a hole in the middle around those little stone piles. There is a nest over there, and that's from a different lineage. And the reasoning why they put those little rocks around, Deborah thinks actually they are trying to do some macro habitats modification, because the temperature and humidity can be different when you put rocks over the sand.
No way.
So maybe they are kind of controlling the temperatures or humidity around their nest. But it is only happen for one lineage. The other lineage never do that.
Wow, that's crazy.
Yeah.
Yeah,
it was really fun.
So cool.
Yeah, it's like so different. It's so not relevant to this project, but also it's kind of relevant.
Yeah.
Yeah.
Yeah, so moving on, I'm curious what, like, after you're done with your postdoc, what's like the dream? What's the big plans for, like, research you want to do, or like what's your goal after graduation from that?
So for the postdoc, it's more flexible comparing to like a PhD. Like PhD, you have to stay there for 6 years to finish your thing.
Yeah.
For postdoc, it's like a short-term contract.
Yeah.
So I renew my contract every year. But because of this harvest ants, dopamine projects going on, I do really want to stay for longer to actually doing more work before I finish this.
Yeah, there's a lot there for sure.
Yeah, because it is for me, it's a unique opportunities to combine the neuron technologies with that social behavior scenario together and look that in the world, like in the natural environments. I want to get more like experience on that. Like for my ideal situation, I'm still really into the collective behaviors in ants. I want to study that. And I really like this idea to look at those social behaviors in their natural conditions rather than highly controlled in the lab. So first, it will be really difficult to mimic those behaviors in the lab. And if we can make those behavior tests or neuron tracing or those techniques works in the wild, it will be much easier to do it in the wild compared to make it happen in the lab. And I think, as you said, it's a really challenge kind of combinations, but at the same time, that is kind of opportunity because we need to make it work. Then this is something unique, and this is actually the things that make me really excited.
Yeah.
So for my future, I'm still really excited about to get my own lab and then can do the things that is combining neurobiologies with study of the social behaviors in ants.
Wow. Yeah, and I mean, you've already done so many things, and there's so many more things to uncover and learn about this. I mean, this was so cool. Thank you so much.
Thank you.
Thank you so much for taking your time to chat with me. But I, for you guys listening at home, I will leave Krisa's information down in the description so you can follow her on all the social media, follow along with what she's working on. I learned so much just talking to you. It was really, really cool stuff. I wish I could talk for like another hour, but
Oh, thank you. Yeah, we can maybe talk later.
yeah, for sure.
Yeah.
Super cool. Thanks so much for doing this with me. Super awesome. I learned a whole lot. I hope that everyone else did as well. It's so cool learning about all this crazy ant stuff and fruit fly stuff and how it all connects to each other. So yeah, thanks so much for joining me.
And thank you so much for interviewing me and giving me this opportunity that can talk about my research.
Of course, yeah.
This is really fun.
Yeah, and I can tell you're really passionate about it. So thanks so much for joining me, and I will catch all of you listeners next time.
And thank you for listening.